Thank you for the nice introductions. So thank you for the organization inviting me to share the research, the progress. Today, so I will be talking about some work just published this year in the evaluation of spiked protein epitopes by accessing dynamics humoral immune responses to COVID-19. Okay, this work is just we published in the Frontiers in Immunology this year. So we know the serious problem is COVID-19, according to the WHO, is almost 600 million people infected and around 6 million people dead because of COVID-19. So if we look at the coronavirus, actually coronavirus divided into 4 different families. Only one family that we cause the very severity of infections, such as MERS, SARS-CoV-1, and SARS-CoV-2. Now, almost 3 years, SARS-CoV-2 is a serious interferes the human life. So if we look at SARS-CoV-2, the genome is around like 30 kb long. So reading from the 14 the encoding regions, so coded for non-structured proteins and the structured proteins, and also with some assistant proteins. So if you deeply look at those the protein the functions, there's some proteins, spike proteins, the glycoprotein, very important. Then envelope proteins, membrane proteins, and also nucleoside proteins, so on and so forth. So we know those spike protein is very, very important because the spike protein is composed of 2 subunits, S1 and S2, in which the S1 protein is related to receptor binding domains. The S2 subunits mediate the viral cell membranes fuse. So very important, the S protein forms large trimer structures, and also S proteins are targeted by host neutralization antibodies. So then we have, if we look at humoral immune responses, the processors, we know that at the beginning, so the immune systems recognize the antigens, then the activation and the proliferation of the B cells. And during these ones, synthesize a secret antibody. So in this case, the most important the early B cells development, the series, they have the change for the antibody, the functions. So S protein is really attract a lot of people, the attention to studies, because this one is closely related to COVID-19 diagnosis, vaccinations, antibody-based therapeutics. However, most study to understand the mechanism for those humoral immune response to the S protein or S epitopes, those were only generated for single technology. So in that case, so a lot of the S epitope recognized only by one approaches. You never can form the integrated view to look at which one is really reflect the epitope, the properties. Another very important view is in the early study, not to focus on designing and not analyzing the patterns for longitudinal recognition of immunoglobulin to the S proteins. That means during the infections, so nobody knows between the processors, those the antibody recognize the epitope with the behavior will change or not. So based on these questions, so we have started our strategies. So our strategy actually divided the two major parts. Number one is we try to collect the patients in different times of the symptom onset. So we collected the problem with like 2 months to collect the serum samples. After collect the serum samples, then we generate different kind antigens according to S encoding the genes. So we make these ones S proteins is expressed like a 14 proteins or truncated proteins. Also, we generate the S-targeted peptide. Another case, we also generate for random peptides. Okay, this one is just randomly from the peptide library. So uses 3 different kinds, the technique, then combined to humoral immune responses. There we can see so which signals truly reflect those humoral responses to recognize the antigens. Then we try to integrate all the information to see what is typically the epitope located. So in that case, we probably collect these COVID-19 patients. We have followed like 19 patients, okay? We collect the time is started for the infections to 2 months. So totally is around like 6 days, 8 weeks to 9 weeks. So first strategy is we designed this one according to SARS-CoV-19, the virus sequencing. Then we express major proteins related for COVID-19. That's recombinant proteins. Then we put it to the microarray. Of the microarrays, then we use the serum to try to recognize the antigens based on the signals. Then we see the immuno signal, how strong and what is located. So look at this one, expression the recombinant proteins for SARS-CoV-2 encoding the genes. We purify the proteins. Most of proteins we also include the GST proteins. That's why easy to detect recombinant protein signals. So based on this microarray SARS-CoV-2 recombinants, so we can see this one, each protein, we have the duplicator, then we can use the immunorecognition, the health, the serum as the control. You can see the second line is negative one, and we can check the immunorecognition for COVID-19 serum. You can see the positive signals. So in that case, we use the GST or just GFP as negative references, and we use the anti-human IgM or IgG as a positive reference. So basically, we look at the microarray, the cluster, or the immunorecognition features to the S recombinant proteins. Either way, we can use the IgMs or IgG. We can see clearly the IgG from the COVID-19 could specifically recognize this SARS-CoV-2 encoding the recombinants. However, for IgMs, look like the recognition capacity is relatively weak. So you will look at compare the IgG recognition signals between health and the COVID-19. We can see all the IgG recognition signals against COVID-19 is higher than health. However, if we look at the IgM recognition signals between the health of COVID-19, except some proteins like against the S1 or M proteins, the IgM shows positive signals. For another non-structured proteins, IgM could not give positive data. Then finally we are looking at the dynamics of IgM, IgG recognition versus total SARS-CoV-19 proteins. From ELISA plots, we can see these IgM recognition signal display higher value at the beginning infections, then gradually decrease of the symptom onset. However, for IgG recognition signals to total viral proteins remained relatively lower at first 3 or 4 weeks, then increase to 4 or 5 weeks after symptom onset. It's very interesting. So approximately after 6 or 7 weeks, the IgG signals started drop down. So that's very strongly indicate IgG affinity against COVID-19, the virus, seems not to last very long time in the human body. So we are looking at the individual recombinant proteins, because we are not the only we make a recombinant protein like S1 protein, we are generate N proteins, E proteins, and so many. So if you look at the IgM signals, we can see the IgM signals of the 2 weeks, so always keep activity lower. However, for IgG against S1, S2, or RBD or N proteins, it looks like the dynamics, the pattern is not consistent. So they leave the questions because those polyclonal antibodies can recognize this S or S2 in the different size. So the dynamic behavior is not consistent to follow the symptom onset. So then, because the full length protein may not completely answer the questions, so we decided to make these the peptide. So this peptide basically we use a different kind of protein, the links. Continuous, we make almost over 200 the peptide. Okay, then the peptides we connected to the BSA, conjugated to BSA, and put it onto a microarray. Then we use the healthy serum, also COVID-19 serum, to make the immunoreactions. So the total is 211 as the peptide conjugate. Okay, we use a triplicate spots for each peptide. Then we use BSA or GFP and the negative control in the anti-IgG, anti-human IgM and IgG as positive control. So in this case, so we look at these, the cluster, right hand is IgG, in the left hand is IgM. So IgM recognition to S peptide is poor specific, but the IgG can specifically recognize some S peptide. It's not recognize all the, okay, just like we have the total of 200. They only recognize like some S peptide. Very similar to the S proteins, the immunorecognition signals either IgG or the IgMs, or except the individual cluster. That means, so different persons, the immune response signal is concentrated cluster together. the implicating the humoral immune responses is really individual dependent. So then we look at those, the S peptide recognized by IgM, IgG is located at different kinds, the genome positions of S genes. So we are clear to see those, the peptide distribution is not evenly distributed along the S proteins. The specific focus at some like regions, like NTD and CDT, FP, and so on and so forth. So then we are looking at those dynamics patterns of human responses. The human responses for different patients, actually the patterns are very different and the recognition is different. However, if you look at the peptide signals, for instance, we have the P3. Now P3 is indicated, the P3 is like IgM. You can look at the beginning and the end. That's consistent, very, very strong. Those are immune different times. They give the very strong immunosignals. So the P8, that's another patients, they can see the patterns very similar. Even talking about IgG is the same thing. So recognition of the humoral immune response to a certain S peptide is relatively stable during the infection the period. Furthermore, well, we found the peptide. Okay, the 7 peptide, of the 7 peptide, the 5 peptide continues recognized by IgM, and all the 7 peptides we are recognized by IgG. Those that behave very similar to the S, the protein, total proteins, the indicator. Those 7 peptides maybe reflect the entire S proteins epitope properties. So after this one, the targeted S peptide, we designed another experiment. This experiment we use what is called AbMap technology. So we use a phage peptide generation systems. We know the phage peptide generation systems. One side is peptide, the random peptide. Another side is the RNAs. So we are first use the antibody, use the patient's serum to enrich those of the peptide. Then we use NGS to sequencing another site. From sequencing the signals, we can find motif of the cluster. So look at this, the data is the generation for motif cluster by antibody binding the epitope technique. So in that case, of the sequencing, we can find which peptide is highly recognized by the patient's serums. So we are looking at this one, total motif cluster per patients, those very variable from 2 and to 11, 16. So very few and only like a motif clusters shared by the tumor, the patient. However, if we are looking at those, the peptides located in the whole, the S proteins, we get a very similar conclusion like a targeted S peptide. That these the motifs clusters, the concentrated located at NDT, RBT, and CDT1, that's regions. Those conclusions are very similar to S peptide result. Fourth one, so we are looking at the dynamics patterns, the human immune responses versus the multiple clusters. We can see the different patients at the beginnings, which the peptide recognized, so we can see it, okay? Then we continue to recognize the whole period. So those motif cluster is really individual features. However, they are the relative stable after symptom onset. So of the three, the technique even like in expression of foldings as proteins, and they use the target, the S peptide, and they use the random peptide, we can put all the information together. Then we do another things, the epitope, the spatial characterized the locations. In that case, we use all the positive, the 40 epitopes from microarrays, and we get another epitope random peptide 26. Then we use DSSP to look at the secondary structures, look at the random coil, the beta sheet, and the olive helix. Then we can use another program to predict the 3 dimensions of structures of proteins. So look at the secondary structures for the peptides. We will see it. So this S peptide and also random peptide, we can see the random coil is significantly higher for all of our helix. Okay, we are looking at the peptide S, the peptide, the random peptide. We can see specifically located these access regions. So in summary, so our data indicates the ELISA or the peptide, the microarray, or the AbMap technique is really useful to study for dynamic change humoral immune responses against S proteins. So the 3 techniques and the different angles indicate the dynamic changes is very important to recognize the variable as the peptide and also indicate for there's a stable for individual the immune characteristics. Thank you everybody for your attention.